Hello, this is Medical Sciences Made Easy by Naphtali Muhumuza, and today we want to talk about the renal, or what we call the kidney nephron. And this kidney nephron, we know that the kidney nephron is the functional unit of the kidney. So the functional cell unit of the kidney is the nephron, and we see we saw in the previous video that we have the right and the left kidney. And we saw how these kidney had nephrons located in the cortex and the medulla. And we saw different types of nephrons, where we saw cortical nephrons and juxtaglomerular nephrons. We saw that cortical nephrons, they lie much within the cortex, whereas juxtaglomerular, they lie more within the medulla. And they are responsible for water conservation. That is juxtaglomerular nephrons. So we want to look at the structure of the nephron, and later in this video we shall focus on glomerular filtration. So, as this is the structure of the nephron, and this nephron, the blood supply enters through the afferent arteriole. And this afferent arteriole is wider than the efferent. It is wider than the efferent arteriole. And then, so blood moves into the glomerulus, then it is drained via the efferent arteriole. And we saw that this efferent arteriole forms the polytubular capillary in cortical nephrons, then in juxtaglomerular nephrons, we saw it forming a vasa-recta. Then, this kidney, or the nephron, it is divided into two, whereby we have the renal corpuscle. Renal corpuscle and the renal tuberous. And the renal tuberous, whereby the renal corpuscle is divided into two. It contains the glomerulus and the Bowman's capsule. So these are the components of the renal corpuscle, which is the first part of the kidney nephron. And then the renal tuberous, that is where the filtrate passes, whereby we have the proximal convoluted tubercle, we have the loop of Henle, which is divided into descending limb and ascending limb of loop of Henle. Then from there, the filtrate moves into the distal convoluted tubercle, and this distal convoluted tubercle is the one that drains into the collecting duct. So this is the two divisions of the nephron, whereby we have the renal corpuscle containing the glomerulus and the Bowman's capsule, and then the PCT, that is the part of the renal tubercle. So after filtration, we see the glomerular filtrate passing first through the proximal convoluted tubercle. Proximal means near the Bowman's capsule. And it is convoluted with many numerous blush borders, then which gives rise into the loop of Henle, which forms the hairpin loop. And this loop of Henle, we have seen, is divided into the thin as descending and thick ascending limb of loop of Henle, which now joins into the distal convoluted tubercle and then to the collecting duct. And this collecting duct is the one that joins to the ureter. Then the ureter takes to the kidney, to the urinary bladder for excretion. So after knowing this, we see in this video we are focusing on the renal corpuscle. We want to talk about in details the glomerulus and the Bowman's capsule. And this is the part we are talking about, which is the renal corpuscle. It is here, whereby the afferent arteriole and the efferent arteriole forming a network of capillary, which we are calling glomerulus. And within the glomerulus, there are cells which are found there. The cells within the glomerular capillaries, together with in between the efferent and the afferent, which we call the mesangial cells. Those cells, we call them mesangial cells. mesangial cells. And these mesangial cells are the ones that are responsible for contraction. They do a big role in the contraction of the afferent and the efferent artery. So that is the mesangial. Mesangial means in the middle of arterioles. So that's why we find it in the middle of the afferent and efferent artery. And apart from that, we see the Bowman's capsule. It has the capsular space. And this capsular space, it has layers. So we're going to see that this Bowman's capsule, it has a layer, two layers known as the visceral layer and the parietal. And these layers, both the visceral and the parietal, they are made up of simple squamous epithelium. So the epithelial lining is simple squamous epithelium. Why? To enable filtration, faster filtration during glomerular filtration. So we are seeing that this is the parietal and this middle one is the visceral layer of the. Then this space in between the visceral and the parietal is what we are calling the capsular space. And we see the glomerulus or the filtration happening within the capillaries and the inner membrane or the visceral membrane of the Bowman's capsule. That is where it is occurring. And lastly, we see on this corpuscle, renal corpuscle, it has again the matura densa. It has matura densa, which is also found in between these arterioles and together with the juxtaglomerular cells lining up the arterioles. So the combination of juxtaglomerular cells together with the matura densa, they form what we call juxtaglomerular apparatus. They form juxtaglomerular apparatus, and this juxtaglomerular apparatus is the one that maintains the pressure. It maintains the pressure within the nephron or within the kidney. So they are responsible for maintaining the pressure. How? By initiating RAS, what we call renin angiotensin aldosterone system, whereby the juxtaglomerular apparatus produces renin and also produces erythropoietin hormone. So we see even hormones from the kidney, like renin and erythropoietin, renin being an enzyme, and then erythropoietin hormone being produced by the juxtaglomerular apparatus of the kidney. So that is the role of the matura densa and the juxtaglomerular cells, maintaining the pressure within the kidney tubules. So after seeing that, we want to see how does filtration occur across the Bowman's capsule. So we are zooming here. I'm zooming it this side. This is the serum forming, whereby I want to show you how the blood from the afferent arteriole is able to be filtered and the rest continues into the efferent arteriole. So when blood is coming, it is containing solutes. It contains, when blood is entering into the glomerulus, when it is entering in the glomerulus, it has components like waste products of metabolism like urea, creatinine, it has ammonia, it has uric acid, it has electrolytes, it has glucose, it has water. We see blood cells like proteins, blood cells like red cells. Then we have white blood cells and platelets. Then we find their proteins. This is the blood entering via the afferent artery. And when it reaches in the glomerulus, we see this blood containing all these materials is going to be filtered. This glomerulus contains the simple squamous epithelia, and this simple squamous have small pores. It has small pores, whereby this is pore, these are the small pores within the endothelium of the epithelial layer of the glomerulus, and these pores, they are the one that we call fenestrations. These are fenestrations, and these fenestrations, they have size less than 0.1 micrometer in diameter. They are very small, and because they are very small, they only allow smaller molecular weight substances in the plasma to be filtered to be part of the glomerular filter. So why do we see blood cells which are big in size, proteins which are big in size not being filtered? Is that they cannot pass through the fenestra. These pores called fenestrations, they are very small, minute in size, 0.1, less than 0.1 micrometer in diameter. So it cannot allow, so it allows only plasma solutes. Those are waste products of metabolism. We see like urea, creatinine, uric acid, ammonia. We see reabsorption of electrolytes, but it does not allow blood cells. That is platelets, red cells, and the white blood cells, because they are big in size. And as the blood which is being filtered continues, it has a second membrane known as basal lamina. This is known as basal lamina, and this basal lamina is negatively charged. It has negative charge. And this basal lamina, it is composed of proteoglycans together with the collagen fiber, and they are the ones that makes it to become negatively, charged. And since it is negatively charged, we see proteins, any molecule except chloride, which is very small inside. Proteins which are negatively charged, they are repelled. So whenever a protein wants to pass through the fenestration, it reaches on the basal lamina, which is negatively charged, and the negative charge repels it. So that's why we see proteins, we cannot find them in urine. So we are saying that this negative charge repels negatively charged proteins. So that is the importance of the basal lamina, which is made up of collagen fiber and proteoglycans, and it is negatively charged. So negatively charged substances like proteins are not allowed to be filtered to pass through the glomerulus because of negative charge. And as it continues, it penetrates through the basal lamina, and it reaches into the cells known as the podocytes. These are the podocytes, and these podocytes, they end into a foot-like, they form what we call foot-like appearances known as pedicels. So these foot-like substances, we call them pedicels, and these pedicels in between, they have filtration pores. These are the filtration pores, and these filtration pores, they are less than 0.007 micrometer in diameter. So any substance, any big substance that must have escaped the fenestration, it is not allowed to pass through the podocytes, because these podocytes, they are end in the pedicels, which are foot-like appearance, and they are very small, 0.007 micrometer in diameter. So they do not allow any big substance to pass through. And those which manage to pass through, they form part of the glomerulus. So we shall find that this fluid which passes through smaller substances like uric acid, like urea, like creatinine, like electrolytes, like glucose, amino acids, some amino acids pass, and water. These form part of the filtrate. These are part of the glomerular filtrate. These form part of the glomerular filtrate. So within the glomerular filtrate, we have this. Then large molecular weight substances like blood cells and negatively charged substances like proteins do not form part of the glomerulus. Why? Because they are big in size, and we have seen proteins are negatively charged, so they are repelled by the negatively charged basal lamina. That's how we can always understand. So after forming the glomerular filtrate, it is this glomerular filtrate that moves via the proximal convoluted tubule into the loop of Henry, into the DCT, and whenever it reaches in the collecting duct, it is now called urine. And we shall see how they are selectively reabsorbed as they are passing through the renal tubules in the next video. Because in this video, we are concentrating on the renal corpuscle. And after seeing how glomerular filtration occurs, we know that GFR, glomerular filtration rate, and glomerular filtration rate, or what we can call renal clearance, is the amount of substance that are cleared off plasma per minute or per unit time. Whereby if it is in a minute, we see GFR in minutes, it is 125 mils per minute. Then it can also be 180 liters per day. That is the glomerular filtration rate. Whereby 125 mils per minute or 180 liters per day. So we see our kidneys in a day, they filter 180 liters of blood every day. That is how great they are. And this GFR is also affected. The filtration across the fenestrine, across the lamina, and across the podocyte is also affected by the pressure, the net filtration pressure. And how do we get this net filtration pressure of 10 millimeters of mercury? Is that within the capsular space, within the afferent arteriole, when it is bringing this blood, it contains water. It contains water. I want to show you how we come up with the net filtration pressure. Because we have seen what affects glomerular filtration. One is the blood flow to the glomerulus. Blood flow to the glomerulus affects. Then capillary, the size of the capillaries. If the afferent arteriole is narrower, GFR is affected. If it widens or if there is vasodilation, so we see the size or the lumen of the vessels also affecting the GFR. And another thing which can affect the GFR is the net filtration pressure. We know that when blood is in the vessels, because of presence of water, it has what we call blood hydrostatic pressure. And this blood hydrostatic pressure is equal to 55 millimeters of mercury. This is the hydrostatic pressure, is this pressure that forces the fluid to move out of the cell. That if I have a blood vessel, this is the blood vessel, the hydrostatic pressure is the pressure, the blood hydrostatic pressure is the one that is forcing the blood to move out of the vessel. So it is a forcing pressure that tends to force the fluid to move out of the vessel. Then the second pressure is the corpuscular hydrostatic pressure. That during filtration, within this capsular space, there is another opposing force. And this capsular hydrostatic pressure, we have seen the plasma hydrostatic pressure is the force that is forcing the fluid to be filtered, isn't it? Then the corpuscular hydrostatic pressure is opposite. So for it, in here, in the capsular space, we have the pressure which is created by the fluid, and it is an opposing force. It is opposing filtration. And this force, because it is opposing, it is 15 millimeters of mercury. That is the corpuscular hydrostatic pressure. Then we know that in the capsular space at the same time there is what we call the oncotic pressure. There is albumin, and this albumin is causing what we call the oncotic or what we call osmotic colloidal pressure. So the oncotic or the plasma oncotic pressure, you can call it oncotic pressure or you can call it plasma colloidal pressure. And this is being created by the presence of albumin in the capsular space, and it is also an opposing. This one is also opposing and is equivalent to 30 millimeters of mercury. So we have seen these two, they are opposing. They are opposing pressures. They oppose. They oppose what? Filtration. Then this one is the one that enables filtration. So this is created by albumin in the corpuscular space. This one is created by the fluid within the corpuscular space. All of them are opposing. This one is enabling filtration. This one is opposing. This one is opposing. So to get the net filtration pressure, you get the blood hydrostatic pressure, which enables the filtration. Then you subtract the opposing, which is the capsular hydrostatic pressure. Even you subtract the oncotic plasma or colloidal oncotic pressure. And we know that the blood hydrostatic pressure is 55 minus the capsular, which is 15, minus the colloidal oncotic pressure created by albumin, which is 30. And at the end, we find 55 minus 45 equaling to 10 millimeters. So we see even GFR, any change in pressure, so long as the hydrostatic pressure falls below 45, we see filtration not occurring. Filtration must be above 45 because we see opposing pressure is 45. So even hydrostatic pressure should be above 45. So we are seeing that the net filtration pressure also affects, apart from the blood flow, other factors is blood flow to the glomerulus. Another one is the lumen, the size of the lumen of the arterioles, bringing in blood, and even the thinness of the glomerulus, of the epithelium of the glomerulus. We said they are made up of simple squamous, and these simple squamous, they are the ones that enable easier absorption because they contain the fenestrations, the basal lamina, and the podocytes, which are enabling the absorption. So this is what we call glomerular filtration, whereby I have shown you even what affect the factors that affect it, and also how we come up with it. Net filtration pressure of 10. Lastly, is we want to look at how GFR is regulated in the body. How do we regulate glomerular filtration in the kidney nephron? And this regulation is enabled by three processes. These three processes, we are going to see they include autoregulation. One is autoregulation, whereby this autoregulation is by both myogenic and tuboglomerular feedback. Then we shall also neuroregulation and hormonal regulation, whereby hormonal regulation will be by RAS and ANP. We shall see ANP and RAS playing a big role in maintaining glomerulus. So autoregulation. Auto means the kidney intrinsically can regulate the pressure or can regulate the rate at which fluids are filtered within the glomerulus. And how? It is by myogenic. The first one is myogenic, whereby myo means muscles. So we see presence of smooth muscles. Presence of smooth muscles in the vessels on the arteriole, that these muscles intrinsically contract. And when they contract, they lead to the reduction in the size of the lumen. And when the lumen size decrease, we see the GFR first increases. It increases GFR because of increase in pressure, but this increase in pressure causes a vasoconstriction. And when vasoconstriction occurs in these arterial vessels, we see it in GFR dropping down. So that increase in GFR first causes increase in GFR. This increase in GFR due to vasoconstriction. Vasoconstriction comes in, and this vasoconstriction causes the narrowing of the arterioles. And when arterioles narrow, they lead to reduced GFR. And that is how myogenic or muscles, smooth muscles, control GFR. Whereas tuberoglomerular feedback, in tuberoglomerular feedback, we see that increase in GFR, it causes increase in pressure. And this increase in pressure, it causes increase in flow. And when the blood flows faster into the nephron, it is bringing more of the sodium ions and the chloride ions. So there is increase in sodium and chloride ions being delivered in the nephron. And these ones are detected by the macula densa, whereby the macula densa, after detecting that there is increase, so we see macula densa detecting increase in sodium and chloride. And this macula densa is the one responsible for producing nitric oxide. So whenever there is increase in sodium, increase in chloride detected by the macula densa, which are found at this point, and this macula densa reduces production of nitric oxide. And we know that nitric oxide is responsible for vasodilatation. So when we reduce production of nitric oxide, meaning the vessels will begin to constrict. So reduction in production of nitric oxide, which is a strong vasodilator, once it goes low, we see vessels undergoing vasoconstriction. And the vasoconstriction leads to reduced or decreased GFR. And when GFR decreases beyond low, we see little blood coming in, meaning little sodium will be supplied. And little sodium detected by macula densa will release nitric oxide. So the mechanism of tuberoglomerular feedback is to decrease GFR in case there is increased GFR. Then neuroregulation, the second one is neuroregulation, and this neuroregulation is done by the sympathetic nervous system, which produces that during exercise or during active exercise, so when you are active, we see sympathetic nervous system releasing its neurotransmitter norepinephrine, and this norepinephrine neurotransmitter, it acts via alpha-1 receptors. And when it goes to alpha-1 receptors, it causes the vessels to constrict. It causes constriction or contraction of vessels, and this one will result into decreased GFR. It will result into decreased GFR when you are active. That's why whenever you are so busy, you are active, you rarely go to urinate. It is minimal. compared to when you are at rest. Because when you are at rest, the GFR increases due to reduction in the production of norepinephrine, which works via alpha-1 receptors. That is what we call about neuroregulation. How these neurons we working via sympathetic, they produce norepinephrine neurotransmitter, which acts via alpha-1, and alpha-1 is responsible for causing constriction of the blood vessels. And whenever blood vessels constrict, they stop or they regulate the amount of blood coming or flowing in. Lastly is hormonal. This hormonal regulation is that the juxtaglomerular apparatus, which we said it is responsible for maintaining pressure of the nephron, produces an enzyme known as renin. And this renin is the one that is responsible to convert a protein known as angiotensinogen to angiotensin 1. Angiotensin 1. This angiotensin 1 is converted to angiotensin 2. Angiotensin 2 by angiotensin converting enzyme. So we see renin converting angiotensinogen to angiotensin 1, then angiotensin 1 is converted to angiotensin 2 by angiotensin converting enzyme. And this angiotensin 2 which is formed is a stronger vasoconstrictor. This guy is a vasoconstrictor. Then this angiotensin 2 which is formed, it can also be converted to angiotensin 3, and this angiotensin 3 can also be converted to angiotensin 4. So apart from angiotensin 2 being a vasoconstrictor, it also plays a big role by triggering the production of aldosterone. That it goes to the adrenal cortex found on the surface of the kidney and triggers the adrenal cortex also to release aldosterone hormone. And this aldosterone hormone is the one that causes much reabsorption of sodium in the distal convoluted tube. And more sodium reabsorbed, more water will also be reabsorbed, increasing the osmolarity. And when the osmolarity increases, it will increase blood pressure. And then the effect of vasoconstriction will also result into reduced GFR. So we see the end result of us reducing GFR. What triggers the macula densa or the juxtaglomerular apparatus to release renin is increased levels of GFR. When GFR is high, it is the one that triggers production of renin. And without forgetting atrionitrile, there is another compound we see under regulation and is known as atrionatriuretic peptide, which we abbreviate as ANP. This one is produced by the atria of the heart. And whenever there is increased blood volume in the heart, that increase in the blood volume, the stretching of the atrium causes production of ANP, what we are calling atrionatriuretic peptide. And this nitroatriuretic peptide is the one that comes into the kidney and causes increase in GFR. So we see among all the regulations, their mechanism is to reduce GFR, except atrionatriuretic peptide, which for it increases GFR. Why? Because what triggers its production is increase in the blood volume. And whenever there is increase in the blood volume, more blood should be pumped to the peripheral tissue. So it comes and causes the vessels, that is the arterial afferent and the efferent, to vasodilate to allow more flow of blood, leading to increased GFR. Thank you so much for listening from the beginning until the end, whereby in this video we have looked at the renal corpuscle, whereby the renal corpuscle we saw it is divided into two. We saw the renal corpuscle divided into glomerulus and the Bowman's capsule. We saw glomerulus is a site where there is filtration, and when we say the Bowman's capsule has both the cell layer and parietal layer, made up of squamous epithelium. And we saw how filtration occurs, whereby we saw the fenestrations, the basal lamina being negatively charged, and the podocytes having pedicels with smaller filtration, enabling smaller molecular weight substance to pass through and not allowing large to pass through. We have seen factors that affect GFR, where we have seen blood flow to the kidney affects GFR, the size of the lumen of the vessels, then the surface area of the filtration surface also does that, then the net filtration pressure also affecting the GFR. And we have ended up by seeing how they are controlled. And we have seen autoregulation by myogenic and tuberoglomerular feedback. We have seen neural regulation and hormonal regulation. Thank you so much for listening. Always remember to subscribe. Remember to subscribe, like the video, and always share your comments. Thank you so much.